Welcome to Mystery Biology. Let's learn something new. In this video, I'll be talking about the principle of Sanger sequencing. Here, the Sanger sequencing, which is also known as the chain termination method or the first generation sequencing. Why it is called the chain termination method, we will discuss shortly. Here, the Sanger sequencing was first developed in 1970 by Frederick Sanger and his colleagues, which is widely used to determine the genetic sequence. And the main application of the Sanger sequencing is it is used to determine genetic mutation which are associated with disease. It is also used in the diagnosis of genetic disorder and to determine the genetic makeup of various organisms. Before going into the detail, I want you to know the difference between deoxynucleotide and dideoxynucleotide. First, let's see the basic structure of the nucleotide, which is mainly composed of the phosphate group, the nitrogenous base, and the sugar, where at 2' position and at 3' position, two hydroxyl groups are present. This is the basic structure of the nucleotide. Now, in case of this deoxynucleotide, here the deoxy means absence of oxygen. That means one oxygen is absent here, and this oxygen is absent at 2' position. But in case of this dideoxynucleotide, where here di means 2 and deoxy means absence of oxygen, that means 2 times oxygen is absent here at 2' position and at 3' position. Why we are concerned for the oxygen at position 2 and 3? We know that DNA is the basic unit of life, which is mainly consists of nucleotides, and these nucleotides are joined together by a phosphodiester bond. And for the formation of phosphodiester bond, 3' OH is necessary. So 3' OH forms the phosphodiester bond with the phosphate group of the adjacent nucleotide. So here the deoxynucleotide has this OH group which can easily form the phosphodiester bond with the adjacent nucleotide. But this dideoxynucleotide, since no OH group is present at 3' position, so it cannot form the phosphodiester bond. Now let's understand this more clearly. Here you can see the structure of deoxynucleotide and the structure of dideoxynucleotide. Here in case of this deoxynucleotide, 3' OH is present, and this 3' OH can form the phosphodiester bond with the adjacent nucleotide. But in case of this dideoxynucleotide, 3' OH is absent, so it cannot form the phosphodiester linkage, so termination occurs. We know the main ingredients in PCR reactions are DNA polymerase, template DNA, DNTPs, and the primers. Here I have shown the basic concept of PCR. First we have the template DNA, which denatures at high temperature, and after the denaturation these two strands separate, and after the separation the primers anneal to these strands, and then extension occur with the help of DNA polymerase. Here the DNA polymerase adds the DNTPs and extend the chain. So now we will talk about the extension step. In case of this normal PCR, with the help of this DNTPs, the chain length increases because the deoxyribose here, it has the 3' OH group which forms the phosphodiester bond with the adjacent nucleotide here, and because of this phosphodiester bond, the chain length increases. In Sanger sequencing also, we need DNA polymerase, template DNA, DNTPs, primers, and one more important thing, which is ddNTPs. But the extension step of sequencing PCR is different, where we use the deoxynucleotide which we normally use in PCR, but here we also use a small amount of dideoxynucleotide, that is DDATP, DDGTP, DDCTP, and DDGTP. All these dideoxynucleotides have unique fluorophore attached to them. Here in the extension step, whenever this DNTPs enter, further extension occur. But when DDNTPs join, reaction terminates. Here the Taq DNA polymerase cannot distinguish between DNTPs and DDNTPs. So every time whenever DDNTPs come, termination of chain reaction occur. This is the reason why it is called the chain termination reaction. Now, the end product of sequencing PCR or Sanger sequencing method is different length of DNA ending with fluorescent labeled dideoxynucleotide. Here you can see different DNA fragments are present. In this video, we have seen the principle of Sanger sequencing. In the next video, we will see how Sanger sequencing works. Thank you.